And we're now moving to our contributor talks. As the first speaker, I would like to welcome Mikhail Pogorelyy, also from St. Jude's, talking about epitope prediction against SARS-CoV-2.
So TCR repertoire sequencing is very exciting, but the issue is that we don't know what vast majority of T-cell receptors we sequence recognize. And one of the major goals in the field is to solve this problem. Someone gives you TCR sequence and you tell them what HLA and what peptide the sequence recognize. And in some cases, we actually could solve this problem. So for example, for this TCR, some of you noticed this very famous beta chain, which is specific for the immunodominant influenza epitope restricted by HLA-A02. And we could do this because we saw this beta chain many times before. It's in many databases, including PDJ-DB. But for novel pathogens, and especially for CD4 T cells, that's not the case. Nothing is in the database yet, but there is still something we could do. And for this study, we combined two publicly available datasets produced with a technique called RTSA. Idea is that CD4 T cells upregulate CD154 molecule, also known as CD4 T ligand, after stimulation with cognate peptide. And in these studies, authors did it with COVID patients. They got PBMCs from them, and they stimulated them with a large peptide pools covering SARS-CoV-2 spike membrane and nucleocapsid proteins. And then they isolated activated T cells and ran them through the 10x genomics pipeline, getting both gene expression and TCR sequences. And two of these data sets combined have more than 100,000 cells, and this is antigen-enriched TCR repertoire data, right? So we have a lot of the cells after we combine this data set, but we have no idea what exact epitopes these TCRs recognize, right? So presumably most of them are SARS-CoV-2 specific because they were activated by SARS-CoV-2 peptide pools, but we don't know exact epitopes. And what could we do to try to answer this question? We need to look what other data is available. All of this data was produced by Adaptive Biotechnologies, by the way, right? So we have three things. First is MIRA class 2 dataset. And basically this is a mapping of 7,000 TCR sequences to smaller peptide pools from SARS-CoV-2. Each peptide pool covers approximately 50 amino acid region. The second piece of data is bulk TCRbeta sequencing from a large cohort of COVID patients. Majority of them are not HLA, so they're not HLA typed, unfortunately. And then there is a very famous healthy control cohort sampled pre-pandemic. So, and of course, vast majority of TCRs in our single cell data set are private. We're only found in one individual. And first step in the analysis is to identify public TCRs in our single cell data using this big cohorts. So, and for this, we just use Fisher exact test and we look for enrichment of TCRbeta in COVID and healthy cohorts, and we identified 1,200 TCRs significantly enriched. So this is an example. This beta chain is found in about 400 COVID donors, but only 23 healthy controls. So I will not go into detail here, but there is an algorithm which allows you to infer HLA types from TCR repertoire sequencing data. And using this, we computationally HLA typed all the donors from the large COVID cohort. And then you could ask for a public clonotype, not only if it's associated with COVID, but if it's associated with a particular HLA allele. And here in this Manhattan plot for this example, you see that this is strongly associated with a certain class 2 allele. Majority of donors have this allele. And then to summarize this public clonotype TCR sequences, we use TCRdist to identify motifs, and this is similarity networks Anastasia showed you before, right? So each dot is a paired TCRalpha beta clonotype. We're connected by an edge if we're close by TCRdist. And colors here show mapping to the MIRA data set. And you see that some clusters are enriched in certain colors, right? And I showed you that we inferred HLA-restriction before. And if you know the mapping to this peptide pool from MIRA, and if you know HLA-restriction, you could then use net-MHC to give you exact epitope. And then we could actually test it. So from this 6 nicely colored clusters, we clone representative TCR to an FAT GFP reporter Jurkat cell line, and we stimulated with a predicted MIRA pool. In one case, we failed to express TCRs. the TCR, but in 5 cases out of 5, we indeed see the reactivity to the target peptide pool. And in 4 cases out of 5, it actually aligned nicely with the predicted TCR, with the predicted epitope. In 1 case, we missed for an interesting reason, but we could discuss it at the poster. So this is how it goes with from single cell data, but you could spin it from other direction using bulk TCR data. And this is with collaboration with Ali Elibedi. He shipped us the samples of TFH cells from lymph nodes of healthy donors vaccinated with Pfizer vaccine. And we did bulk TCR repertoire sequencing. And again, we do similarity network. And here the color means the donor. And in TCRbeta, you see some clusters, not big, shared between donors. But if you go to TCRalpha, you see this gigantic motif, which was shared among all donors. And we were interested what this T cells recognize, but we have only one chain, TCRalpha. And now we go back to this single cell TCR data, and we look up for the TCRs which have this alpha motif, and we pull beta chains from them, and we look up this beta chains in MIRA dataset, right? So we use single cell data as an intermediate, and we get a lot of hits to a certain spike peptide pool, and then we use predicted HLA restriction to narrow it down to a certain epitope. And once you have HLA restriction, once you have an epitope, what you could do next, you could ask your great Australian collaborators to make an HLA class 2 tetramer. And this you could actually use to track CD4 specific for SARS-CoV-2, both in peripheral blood and in fine needle aspirates. And what's interesting here, but we see very different dynamics, although it decays quickly in peripheral blood. In some donors in the lymph node, it is stable on very high level for more than 6 months. And with this, I want to thank all the people who participated in this work, especially Anastasia, who now is in the audience, and she's a co-first in all the stories. And I am happy to take any questions and chat with you more at my poster later today. Thank you.